Hello everyone. In this class, we'll learn about muscle energetics. Adenosine triphosphate. Adenosine triphosphate is the immediate source of energy for contraction in skeletal muscles. During ATP hydrolysis, the terminal high energy phosphate bond of ATP split to form ADP and phosphate, with a release of 7.3 kilocalories for each mole of ATP hydrolyzed. So this reaction showing breakdown of ATP, where it gets converted into ADP and phosphate, releasing 7.5 kilocalories of energy. The functions of ATP are as follows during muscle contraction. It supplies energy required for cross-bridge movement during muscle contraction. ATP binding to myosin breaks the actin-myosin interaction and allows the cross-bridge cycle to continue. It provides energy to the calcium magnesium ATPase pump that transports calcium back into sarcoplasmic reticulum and initiates relaxation. The sodium potassium pump in the sarcolemma utilizes the energy derived from ATP hydrolysis to maintain the membrane excitability. In resting muscle, less ATP molecules are present in the sarcoplasm. Once the contraction is initiated, ATP store can sustain contraction for fraction of a second. For contractions to continue, the muscle requires an enormous supply of ATP which has to be generated immediately. ATP is regenerated by the addition of one of the phosphate group to ADP by three ways. First one is phosphorylation of ADP by creatine phosphate. Second one is through oxidative phosphorylation of ADP in the mitochondria, and third through the phosphorylation of ADP by breakdown of glucose to carbon dioxide and water. Second one is creatine phosphate. Creatine phosphate is also called as phosphoryl creatine. Phosphorylation of ADP by creatine phosphate is very rapid, non-oxidative process which provides ATP by transfer of a phosphate group from creatine phosphate to ADP catalyzed by the enzyme creatinine kinase. The concentration of creatine phosphate is about 5 times that of ATP in the resting muscle. The above reaction is stimulated by a fall in ATP and a rise in ADP concentration. The amount of ATP that can be formed is limited by initial concentration of creatine phosphate in the sarcoplasm. The ATP supplied by this mechanism sustains the contraction for few seconds. During recovery period, the concentration of creatine phosphate is restored back to the normal level by addition of ATP formed by glycolysis to creatine. Also, some mitochondrial ATP transfers its phosphate group to creatine. As the levels of ATP and creatine phosphate fall, the other mechanisms are activated to provide ATP to the contracting muscle. At rest and during light exercise, muscle utilizes free fatty acids as its energy source. During moderate to heavy exercise, metabolism of glucose and glycogen has the primary role to generate ATP. In the presence of oxygen, aerobic glycolysis takes place, the end products being carbon dioxide, water, and 38 molecules of ATPs. If oxygen supply is inadequate, anaerobic glycolysis delivers lactic acid and 2 ATP per molecule of glucose. Oxygen debt. During rapid strenuous activity, even with a high respiratory rate and maximum possible dilatation of the blood vessels, ATP generated by aerobic glycolysis is not rapid to meet the body's energy need. In that case, anaerobic glycolysis occurs to fulfill the ATP demand. For example, when a person runs for an hour, around 95% of the energy spent comes from the aerobic glycolysis and only 5% from anaerobic glycolysis, whereas a 100-meter dash completed in 10 seconds, 85% is derived from the anaerobic glycolysis. After the exercise is over, body has to accomplish the following functions: convert surplus lactate into pyruvate and replenish the ATP and creatine phosphate stores in the muscles, and to replace oxygen that came from myoglobin. For all the above functions, oxygen is required. Therefore, even after the termination of exercise, the respiratory rate remains high for some time according to the severity of the exercise. The amount of extra oxygen taken up by the body above the basal oxygen consumption following exercise is known as oxygen debt. Rigor mortis. During cross-pitch cycle, binding of ATP to the myosin head breaks the actin-myosin interaction and brings in relaxation of the muscle. If cellular energy stores are depleted, as happens after death, cross-bridge detachment cannot occur due to lack of ATP. Therefore, myosin heads remain in the attached state. Following death, cytoplasmic calcium concentration remains elevated because of the following reasons. Calcium is not pumped back into the sarcoplasmic reticulum in the absence of ATP, and calcium diffuses from ECF into the cytoplasm. This is because after death, the muscle membrane becomes inactive, which cannot maintain high gradient of calcium between ECF and ICF as it occurs in life. The inactive membrane of sarcoplasmic reticulum cannot hold back calcium, which diffuses out of the sarcoplasmic reticulum into sarcoplasm. Raised cytoplasmic calcium concentration initiates the contraction by exposing myosin heads on actin and permitting the formation of cross bridges that do not detach due to the lack of ATP. This leads to stiffness in the muscle, which is known as rigor mortis. It starts about 2 to 3 hours after death and gets completed in about 12 hours after death. The stiffness disappears 48 to 60 hours after the death due to the disintegration of muscle proteins. Thank you.